Is lab-grown meat cultivated using cancerous or pre-cancerous animal cells? No, that's not true. Cell-cultured meat is cultivated using animal cells that have been modified to replicate indefinitely. Cell-cultured or lab-grown meat is regulated in the U.S. under the same stipulations as conventionally grown and slaughtered meat, poultry, and seafood. Furthermore, regulatory agencies do not allow for cancerous animal meat, whether cultivated in a lab or conventionally slaughtered, to enter the U.S. food chain. In June 2023, the U.S. Food and Drug Administration, FDA, and the U.S. Department of Agriculture Food and Safety Inspection Service, USDA-FSIS, deemed meat produced by cultivated poultry cells as safe to eat. The claim originated in a 152-minute video posted on TikTok on June 25, 2023, with a text overlay that read, "What do y'all think is it, cancer-made meat?" A caption that accompanied the video read, quote, "Replying to at ReciCup 921 #greenscreen video at Kenny the Trainer #man-made meat #fake meat #what do you think #foodie TikTok" end quote. Below is how the post appeared on TikTok at the time of this publication. Source: TikTok screenshot taken Tuesday, June 28, 20 hours 32 minutes and 0 seconds Coordinated Universal Time 2023. The logic presented in the video falsely suggests that because lab-grown meat uses fast-multiplying cells, and cancer cells multiply quickly, that lab-grown meat must be produced from cancer cells. This is not true. Cell-cultured or lab-grown meat is not produced with pre-cancerous or cancerous cells, but rather with immortal animal cells that have been modified to repeatedly replicate. Furthermore, animal meat containing cancerous cells is not allowed to enter the U.S. food chain, and lab-grown meat is subject to the same statutory requirements and regulations as conventionally grown meat derived from slaughter. The video shared to TikTok included authentic clips from a reel published to Instagram by the Associated Press on June 24, 2023, that described the process of lab-grown meat as cells that, quote, "come from a biopsy of a living animal, a fertilized egg, or a special bank of stored cells. Companies choose the cells most likely to reproduce quickly. Then, they're placed in tanks in a bath of cell food designed to give them everything they need to grow. Inside these bioreactors the cell can change into types we normally eat, including those that make muscle, connective tissue and fat. And the cells multiply over and over and over. After several days or weeks, the meat is removed from the tanks to be shaped into cutlets, hot dogs, nuggets and sausages. Then, it's ready to be cooked, served and eaten." End quote. Cultivated cells are not made from cancerous or tumorigenic animal cells. The Guide to Research Techniques in Neuroscience reports that immortalized cells replicate indefinitely in one of two ways: either spontaneously, such as cancer cells, or by artificial manipulation. In a memorandum released by the FDA on November 14, 2022, the agency described in a public document that cell material is comprised of cultured chicken cell lines from the Gallus gallus species that are originally isolated from either adult chickens or mid-stage fertilized chicken eggs. These cell lines are adapted to suspension culture, which allows cells to function and multiply, and then immortalized to proliferate indefinitely through either a selection in culture or through the introduction of chicken telomerase reverse transcriptase, TERT, a gene manipulation that enables the cell to continue replicating. Cells are then harvested in the form of sheets of cells washed from the surface of the culture vessel into a collection basin. These sheets undergo subsequent washing and are adjusted for moisture in a temperature-controlled environment. Harvested material is a coherent tissue of chicken cells, similar in composition and nutritional characteristics to conventional poultry products. The FDA describes this process in more detail. Quote, "Step 1. Manufacturers typically start with a sample of cells from the tissue of an animal, a process that does not require harm to or death of the animal. Some cells from the sample are selected, screened, and grown to make a bank of cells to store for later use. Step 2. A small number of cells are taken from the cell bank and placed in a tightly controlled and monitored environment, typically a number of sealed sterile vessels of increasing size, that supports growth and cellular multiplication by supplying appropriate nutrients and other factors.